Roger that.
And
Pretty
it's
constant.
creeping towards the ship, if I'm not mistaken.
Understood.
Bobby's down there.
Both of those things are expected.
All right, I gotta get my heading around here. There we go.
There you go, there's bottom.
Bottom. All right.
RV's
Back
on
in
bottom.
business.
Are you ready for DVL? Sorry,
You can do, sure.
switch you back to this. And I'll do a reset.
Yeah, arbitrary. I'm still moving.
Okay. Heck is it? Okay. Ready?
Yep.
Okay. That's where you're at right now.
Thank you.
Argus is
30 meters.
Yeah. 30 meters off the wall.
It's pretty constant. That one to the last one was pretty good, so
Yeah.
should be okay.
It's moving a bit closer. Mostly along contour, but slightly up. So we'll see you creeping in.
Creepy. I wonder if I can actually drive forward now. Let's see.
Perhaps. There you go.
Any sign of life yet?
You know, Rennie, that's an argument for working the spine of the ridge, you know?
Yeah.
Yep, absolutely.
Always in high high wind and current states, being on either a spine or the slope that's that you won't crash into is ideal.
Okay,
That could have gone a very different way if we were working the other side of the slope.
let's take this wrap out before watch change. So I'm gonna get in front of you here, and then you can just follow me around as I turn around.
Doing a modified turn off
Full on
all the way around.
victory lap.
Are you doing that just by turning, or are you driving ahead too? It looks like both.
I'm driving ahead. Keeps
Yeah.
the tether streamed out behind me.
Ah, got it.
Now I think we'll be going 070 if we want to set them up.
Yeah, it looks
070, roger that.
Yeah,
Yep.
something like that. Depends on where Argus finally settles.
Sounds good. We can plan for that. Are we looking that way? So keep going this way.
Complete the loop.
Getting there.
Complete the loop.
Oh. Wow. Yeah.
All right, cool. While you follow me around, you can start coming up, because I'm going to start coming up too. And we can do regular Delta operations.
Are we coming up a bit?
You're coming up, yep.
Okay.
Yeah, way better.
We're just going to get this sorted and hand it off.
We can do once you get up and ahead and you want to hold position for a bit, we can try to hone in on a reset.
Hey.
We're now conducting watch change for the 12 to 4 watch.
Video's off comms.
I hit this button. Perfect. So we are going. Okay, the ship stopped. We were moving 80 degrees, which like if we want to go to waypoint 2, we should change slightly our bearing. Change it to 55. Does
Hey, good afternoon, guys.
Hey, everyone.
Good afternoon. Yeah, that sounds good, Albert.
Bridge nav. Can we change
We
our
still got-
bearing to 55 degrees, please? And also steps size 30 meters.
Sounds like people are still getting a little settled. Is everyone in science here?
Thank you.
Yeah, we're still kind of getting settled, but everyone's up here.
Cool.
We want to step.
Cool, cool.
Yeah.
So bridge nav, step please. Thank you.
So, Mike, are we back to full capabilities here?
Yeah, we're good right now.
Okay.
Yeah.
Cool.
Yeah, ship's moving already. We are going towards waypoint 2.
Great.
Feels like it's been a while since we've all been here.
Yeah, they got 3 rocks this morning.
Did they? That's nice.
Okay, everybody's here now.
I think we're all here. Do we want to do a round of introductions since it's been a while since we've all been in the van and we probably have some new viewers? Maybe start, who do we have in science today?
I am Sam Georgian. I'm the watch leader today from Marine Conservation Institute in Seattle. And I have a background in deep sea corals, and then now I mostly do ecological modeling.
Who else we got back there? Sarah, you there?
Yep, I'm here. I'm Sarah, and I am getting my PhD in oceanography from Oregon State right now. And I study ecosystems in the deep sea and shallow seas, looking at how microbes and animals and ocean chemistry interact to form ecosystems. Your turn, Dwight.
Wasn't paying attention. I'm Dwight Coleman. I'm sitting in the Science 1 position, or Science 2 position, whatever we're calling it, between Sarah and Sam, and helping out on watch with some of the geological observations and picking up rock samples, and just making general observations and helping the dive progress on schedule. And at home, I'm the director of the Inner Space Center at the University of Rhode Island Graduate School of Oceanography.
Awesome. How about front row? Albert, you want to tell us about you?
Yes. Hi, I'm Albert. I'm the navigator for this watch, which means that I try to get the ship in the right position so the ROVs can do whatever the science need them to do. And at home, I work at an underwater robotics lab in Girona, at the northeast part of Catalonia. And yeah, that's it.
Awesome. How about ROVs today?
Michael Hannaford, Herc pilot, from St. John's, Newfoundland. 4th year out here. There we go.
I think you're muted. There we go.
I'm Gabby. I'm sitting in the Argus seat. I'm from Rhode Island. I'm taking gauges right now.
Busy. Sorry, Gabby.
That's all I knew about myself at the moment.
You're living in the present.
Yeah,
Good.
exactly.
Good.
Bridge nav, step again,
Just
please.
living very much in the present.
I'm Nick. I'm the video engineer on the watch. I have a
Could do a zoom on this guy, please?
relatively simple job. I just make sure the picture looks good and the pilots can see where they're going. I'm from Third Rock from the Sun. I hear it's a pretty hot place these days.
Nick, can you zoom in there?
And I'm zooming now.
Sorry, I'm distracting you from your
Whoa.
job.
No, I don't think so. Yeah, it's on 100% now.
That's interesting. The like 2 colors.
Yeah, I think it maybe is another Corallidae, but colonized by zoanthids.
Do you think it's the skeleton that we're seeing poking through some?
So you think
No,
that the
'cause
orange things are
There's
different?
polyps, yeah.
Yeah, there's polyps, so it's not like dead skeleton. No, I think it's just I think the pink are colonizing zoanthids.
What is that green thing?
A sponge, I think.
The sponge
Where?
growing on
Oh.
the top
On
there?
top.
Or a tunicate or something.
Or an egg case, but we've definitely narrowed it down.
Yeah. It's not a rock.
Not a rock.
It's not a fish.
Yeah, is that the tightest zoom we can do on that?
You go in more, Nick?
Yeah, I can do tighter, much tighter.
Because a lot of things do lay egg cases on these corals, but
Nice, Mike.
Wow. I mean, it kind of looks like a rock, except that it's not.
I wouldn't say it's a sponge anymore.
Huh.
no, and
I've never seen an egg casing that looks like that, but it does resemble what egg cases normally look like on things.
yeah, I mean egg cases are tough because there's
Yeah,
just like
so many different types.
so many. Yeah, I'm not sure. I mean, it doesn't look like any of the cat shark or
Or snail
octopus ones that I've seen before, but that's all right. I think we can move on.
Wide there.
That's interesting.
Cool.
This rock looks cool.
Did you introduce yourself, Kelly?
I didn't, thank you. My name is Kelly. I'm a science communication fellow on the Nautilus. I'm from Washington State, and there I work for the government, actually, Department of Ecology, and I do a lot of science communication work around nutrients and different pollutants and how they impact ocean acidification in Washington specifically. And this is my second time on the Nautilus, so super happy to be here.
Another step, Mike.
Yeah, sure. Yep.
Can we just do a
Breathe
quick
snap.
zoom on this guy and the
Step again,
lasers
please.
right now?
Yeah, go ahead, Nick.
Chrysogorgid. Oh, yeah.
How about the Brussels sprouts right behind it?
Where?
That's what I was going for.
Yeah, the Brussels sprouts.
Mm.
You'll see.
You'll see.
You'll
You'll
see.
see.
Just wait for it.
Is that the common name?
Yeah, that's the common name that I just invented.
Brussels sprout.
There's an algae called sea cauliflower because it looks exactly like cauliflower.
Oh,
And
really?
I love that name so much.
What do you think this is, Sam?
Chrysogorgidae.
So,
It has something living in
Sam,
it.
the root of a lot of these corals' names seems to be gorgy or something like that. Do you know what that means?
Steve was just talking about this in the chat earlier. It
Oh,
says
nice.
it refers to the Gorgons of ancient Greek mythology.
Oh, no kidding. So it's not even a Latin base.
Go wide
Not even
there,
a Latin
video.
root.
Now, why they named them after that, I don't know.
Brussels sprouts, I'm telling you.
Oh, yeah, they said they were seeing some interesting sponges down here.
I see it. Yeah.
Is that how Brussels sprouts grow?
Oh.
They grow on a stalk.
Interesting. So the Gorgons were 3 monsters in Greek mythology. And the most
Can we get
famous
the
one
other
of
set?
them was
Zoom in there?
Medusa.
Yeah, go ahead.
Yeah, she had snakes for hair, so
Yeah, kind of coral-like.
Interesting.
yeah.
I do know the Greeks were very confused about the corals they found. They, at various times, included them in kind of their rock description books and their flower
Oh, that's
Huh.
awesome.
That's
description
really
books.
At the
cool.
same
they
time,
were-
or
No,
Look at
kind of
this
different
Brussels
times.
sprout stalk.
Yeah.
Good call, Gabby. I see that.
So if you've ever asked yourself, I'm not really sure if corals are plants or animals, you're in actually very good company.
way better than if you think that they are rocks.
Right. Well, still
Okay,
good
full
company.
wide.
Sarah, do you know who the other 2 Gorgons were besides Medusa?
Yeah, but I just exited.
Oh,
Hold on.
sorry.
Wait,
I
wait,
can pull
one
it
was
back up.
Medusa?
One
Yeah.
Yeah, one was
Medusa.
Medusa. So
That's how we call jellyfish in my language.
their names were Stheno, Uriel, and Medusa.
Oh.
The daughters of Echinda and Typhoon. Echinda, that's like
Echinda?
Echinoderms? Yeah. It's a sea star, yeah.
Caesar and cucumbers. And can we zoom on this whip, please?
Curious.
That's very cool.
Zoom on which one, Sam?
The whip in front of us.
Yeah.
Apparently Echinda and Typhoon were the mother and
Okay, go
father
ahead,
of all
video.
monsters.
It's interesting. It has like some
And
branching.
Typhoon,
So it's a bamboo coral, but I'm
Storms.
not. Huh. Usually the whips you don't see with any
It's good right now.
any branching.
So it gets up in the red, but it's
I should say usually I don't see. It's not like this is probably new to science. or anything.
new to my personal perspective.
Yes. That's good, Mike. Thanks. I think they sampled one of these earlier.
okay. Full wide.
So how full are we on samples?
We have lots of space. Lots of space.
All right. We're also the last watch of the dive again, so
Oh, tomorrow morning. Wait, this
Tonight, you mean?
tonight?
yeah.
No, that will be tomorrow morning, actually.
Yeah, it will be.
Oh, yeah.
I did doubt myself there for a moment.
So we can't completely play the game of we're just let the future watches worry about where they're gonna stick stuff.
Because that's us.
Yeah. But we have only 2 bio boxes have things in them. And this 2 of the slurps are full, but apparently the last slurp may have clogged a little bit.
Yeah, Emil mentioned that. He said they slurped something after though, and it still worked.
Yeah.
Mushroom coral.
Wow, there's lots of little corals up here.
Oh, wow.
And
We zoom on this big guy just below the lasers, please.
Yep. Go ahead, video.
Another
pretty big
bamboo.
guy.
Yeah, it is. I wonder if it's the same as the other one we saw, just way more branching, or different.
Do you want to keep moving or do you want to wait 1 sec?
No, keep moving. I'm gonna go back up there now.
Bridge nav, step again, please.
Full wide, please.
Bridge, this is nav. Step again, please. Thank you.
so, Dwight, how do we pronounce this type of seamount we're on right now?
Well, I say Gio, but some people say Giot. I don't.
Gio or
think it's
Giot?
I think it's Gio,
Gio.
like with a silent T at the end.
It's French, right?
Yes. So I would say Gio is the proper pronunciation.
Gio.
Gio. And how is this different from the other seamounts we've been exploring?
Well, the theory is that this has the shape, the morphology is completely different. So it's got a completely flat
That
top.
guy's cool.
Unlike some of the other seamounts that we've been diving on that have more
Zoom in there, video.
rounded or pinnacle-shaped top to it, summit to it.
Huh. So it kind of plateaus out.
Yeah, and they generally stand alone by themselves and rise up out of the seafloor as a sole seamount and not part of a larger like ridge or adjacent to other sort of large seafloor formations. And the theory is that they were formed, well, they have a volcanic core to them, and they formed at much shallower depths, and then have since subsided down to the depths that they're at here, and that the top of the Gio was basically denuded or eroded away at sea level.
Yeah. Oh, interesting.
And so it must have lived at sea level for quite some time to erode down that easily. But it could also be a function of the basically erodibility of the basaltic rock that forms its core and the coral formations that live around the surface of it. So there's a lot of other factors that determine the shape of the Gio.
Are we planning to explore like the top of this seamount?
Yes.
Cool.
By the end of the dive, we should be up on the top
Zoom
of it.
in there, video.
Very cool.
And hopefully we'll have time to do some work up on the top.
Huh. Are they common Gios?
Yes, they're
Okay.
they're all over the Johnston Atoll region, Central Pacific region. Some are quite large. This is actually a smaller one, so it's kind of interesting.
What's the diameter?
I think it's like 5 miles across.
Oh wow, really? That's way bigger than I was imagining.
I haven't really looked at the multi-beam we got yet.
Full wide.
The multi-beam lines that we were doing to collect the data down to 5,000 meters on both sides were like 12 miles or so.
To get to the full, yeah.
To get to the 5,000
right.
meters depth. Which
So I
is
think
not
the very
full depth.
summit, the red part,
Yeah,
is
that's
really a
like,
couple
like
miles.
half of it. No,
Yeah.
it's slightly more, I would say.
Is
yeah.
it 6? 5 or 6?
Yeah, yeah. A third. Definitely one
So
third.
that is pretty large, actually.
Yeah.
But there's other geodes
What
that
bearing
are
are you doing, Albert? 70?
many tens of miles.
What?
What's the
across
ship's
the
bearing?
top.
It's 55.
Because you can
Point
imagine
Argus
like what's
over there
happening
for a
at
minute.
Hawaii now, as those islands sink deeper and deeper and subside. You know, they're huge islands, right? So some geos are the size of like one of the Hawaiian islands.
Wow.
And how do these mountains form? I was looking other things.
How did it form?
Yeah.
The theory is that it formed at sea level, that it erupted
Mike,
and,
did you catch
and
some of those things that
came up
Trevor was
above
saying?
sea level and then was eroded
Yes.
down to sea
Okay.
level
Okay.
and then subsided on the plate as it moved away from the hot spot and it moved away from the mid-ocean ridge system.
Zoom in there, video.
That's what I thought yesterday night when we were mapping it. It's like that could potentially be.
Oh, that's gorgeous.
That's really beautiful. We haven't seen that many that like stalk out like that.
Yeah, we're seeing like a lot of diversity in the sponges on this dive already. Instead of just that kind of glass base sponge that we've been seeing everywhere.
Yeah, this is really cool.
Yep.
Bridge nav. Step again, please. Thank you.
I'm not quite at 55 yet, Mike.
Yeah. Full wide, please.
Albert, can I ask how far is it to waypoint 2? And then how far is waypoint 2 from 3?
The waypoint 2 thing, it's on your screen. You can have it if you want. It's 373 meters away from Hercules. And to waypoint 3, you don't have it. I have to look for it.
Okay, my goal heading is 53.
Roger.
We have to go through this bit to search for rocks.
Right.
Zoom in there, video.
It's roughly
Oh, wow.
1500 because I guess we have to do like this
1500 from us or from waypoint 2?
from us.
Okay.
From waypoint 2, 1000.
Okay. Thanks. So we won't quite make it there on this dive. Oh, that's beautiful.
That's really beautiful. It's neat to see this one. It looks like it has like holes going through it, like so porous.
It's like some kind of bubble wrap.
Yeah.
Hey, if you're breathing into the mic, can you just mute your microphone, please?
Okay, full wide, please.
Another step, Mike.
Yep.
Bridge nav, step again, please. This DVL is behaving like crazy today. What's your salinity and sensor speed and sound speed down there?
Salinity's 34 and 1493.
So Sam, I know we've talked in the past about like the coloration of sponges down here, and they're all kind of white. Does this serve as a functional purpose? Is there a reason they maybe don't have any coloration in their tissue down here?
So these are glass sponges, so I'd say it's just a function of what they're forming their skeleton out of. A lot of the sponges we see that have color, we think are their secondary metabolites that they're using for like defense and things like that.
And so they may not like need that down in the deep sea as much, potentially, or?
Well, I'm not a sponge biologist. A lot of the colorful sponges I think are more like demo sponges, so not in the glass.
Zoom in there, video.
Glass sponge group. I'm not aware of any glass sponges that have a lot of color.
Yeah,
Sarah?
that's what I was thinking when she asked the question. That I just thought it was because so many of these are glass sponges that we're seeing.
Yeah.
And yeah, I can't think of any glass sponge I've seen with color.
It's really beautiful when you get like a whole reef of glass sponges. Like there's a few off the coast of British Columbia.
Oh, cool.
Just like fields of glass, basically.
Wow.
Yeah, I've worked there a few times. With,
oh, cool.
Yeah, that's amazing. It's cool
That
stuff.
sounds awesome. Wow, this one's interesting too.
The sponges get absolutely enormous too.
Really?
Just like giant tubas.
Tall or wide or both?
Everything.
Both.
Huh.
All of it. They flare out just like hugely.
Cool.
Wow, that sounds awesome.
Big tubas and trombones and sousaphones.
Okay, full wide, please.
Another step.
We're looking okay still.
Yeah.
Bridge nav. Step again, please. Yeah, this is going crazy.
Why do you suppose that is? Why is our USBL so bad?
I don't know. Because we are not in a cave or like, or we are on a ridge. It should be
Zoom in there, video.
pretty good.
I wonder if there's like some density stratification that's like much stronger than it was where we were last. It's just sounds just bouncing around.
But it was not that bad, like
Or
5
when you
minutes
guys
ago.
looked at the, oh really?
Yeah.
Huh. Yeah, that's really bad.
Yeah.
Okay, come on.
Mike, would you prefer if I just kept the camera in one position? So you
Uh,
can tell where you are?
I
or-
don't care. It's
Okay.
whatever.
So we have a question from actually a
Getting
former
a little
science communication fellow
Creeping up on that
who's watching with his kids right now.
hillside.
And they're wondering if we're likely to see any animals down here, but they also want us to know that they know corals are animals. So one step ahead of us. But if there's any kind of bigger...
And sponges.
There we go. And sponges. If we're likely to see anything else down in these deep waters.
Well, we have seen on our watches previously in the cruise, we've seen a shark.
Nobody was
a couple
interested in that.
sharks. Yeah, sharks are wildly unpopular. Several octopuses, although we haven't seen one in a while.
No, but we were on a roll there. We were seeing
know.
like
Oh,
three a
octopus.
dive.
I mean, we saw the best. Was it?
We did.
It was an octopus after, right?
That's
it ended
what.
up being an octopus.
Yeah, that's
Yeah,
what
it was,
they have
yeah.
determined.
Sort of like split the middle between a vampire squid and a Dumbo octopus.
Yeah,
Yeah.
that thing was
Hybrid.
amazing.
Yeah, that
Another
was
step, Mike.
one of the coolest critters I've ever
Yes,
seen
please.
Bridge
underwater.
nav. Step again, please.
a position previously held by the vampire squid and now held by the vampire octopus.
And if they go on to the Nautilus, well, I guess they're on the Nautilus website. I think they just posted the Deep Staria video, right?
Yeah, they did.
It was a really cool highlight of a jellyfish from a few days ago. Not on our watch, unfortunately, but we can't take them all.
True. And we've been seeing a lot of fish, although we haven't seen many fish today.
No.
Or any fish today, actually.
Sun must be very busy. Oh, it's flattening out here.
Yeah, we're at the top-ish. Top-ish.
I think there's your Brussels sprouts again, Gabby.
Where?
To the right. See that on the right? Is that the same
Oh yeah,
thing?
look, more
Little
Brussels
ones.
sprouts. Little baby Brussels sprouts.
Oh, I meant to look this.
It's one of my favorite vegetables, actually.
Me too. I love
that's
Brussels
a cool
sprouts.
one.
Oh,
That
look at
sponge
that.
further over, right?
Stocky sponge.
Yeah, that is cool.
Do a zoom here, video, please.
Brussels sprout zoom.
This one's having a hard time or something.
These Brussels sprout ones both looked kind of dead-ish,
Yeah.
and I'm just going kind of by their color, because like.
Oh.
Yeah.
Yeah,
This is how they grow.
Sam's back here Googling a picture of a Brussels sprout stalk.
Okay, full wide.
Gabby is 100% on it.
I know my Brussels sprouts. They're
Yeah.
literally my favorite vegetable. Besides sweet potatoes. But I haven't seen a sweet potato sponge yet.
That
So I
would
s-
be like a sea cucumber.
I miss sweet potatoes.
I studied abroad in Australia, and we stayed with homestay families. And the first night, my homestay mother asked if I liked Brussels sprouts, and I was trying to be polite, so I said sure. And guess what I had every night after that? She was like, he just loves Brussels sprouts, can't get enough of them.
I don't even know what that is.
So I take it you don't like them?
No, it's very safe to say that I don't like.
Depends how they're prepared, I think.
Yeah, that's a little mushroom
Yeah, if
coral
you've
down
had
there.
the old world version of, boiled in a
Boiled.
pot until that's
Until
pretty
they're
bad.
squishy.
But you
But
can
if
have
you
them.
get them like, they're just baby cabbages, right?
Yeah.
They're
Full
delightful.
wide video.
I've had this one Brussels sprout recipe before that was so delicious. It was Brussels sprouts like marinated in like a Dijon mustard sauce and then roasted in the oven.
Yeah, there's
so
lots of
good.
great things you can do with them.
Guys, this is a dangerous conversation.
Just not boil them.
Don't let it
They
escalate.
smell when they boil.
Yeah, we are getting to the point in the cruise where there's like not that many veggies left. Which coincides precisely with this time that you start talking a lot about vegetables, and I start seeing them underwater.
In all of the sponges.
In all of the sponges.
Is that kale?
It looks a lot like kale.
There's a little over a week left. We'll see what's left for vegetables in about 5
We're
or
still,
6
they're doing a
days.
pretty good job. We still got like tomatoes and cucumbers and peppers.
Only 1 week left?
Over 1 week.
Oh,
Let's not get excited.
As long as we're good on French fries.
Oh, we're always good on fries.
And tots.
Yeah, we'll always be
Let's
good
be
on that.
serious. It's all about the tots.
So I guess I've been talking about it way too much because my wife messaged me this morning. She was like, "How's your day? It's ice cream day."
Oh, is it ice cream day?
Yeah,
Wait, is it
It
really?
is
Wow.
ice cream,
nice.
ice cream Sunday.
Sunday already.
I guess it's ice cream for dinner because it was not there for lunch.
Yep.
Ice creams for dinner.
I'm not good at making observations like how regular things happen, like we'll have ice cream on board and this kind of thing. Like,
Do you know what day it is, Albert? That's the question.
yeah.
I mean, I
Kinda.
almost never know what day it is out here.
He knows where the ship is, but no
I know.
idea.
I know where the ship is and that the ship needs a step. Please,
That's the ship.
step, please.
Speaking of which,
Now that we're at the top of this, do we want to iron out the layback on Argus?
We have to wait to go all the way until waypoint
Yeah.
2, so I was planning on doing it just before
Oh
that.
yeah.
Okay.
I think now we don't really worry about it.
Could you zoom on that big white guy, please?
Roger. Okay, video, zoom in.
Oh, that's really nice.
Wow.
There's a gramophone.
Oh, yeah.
Wow.
it's a satellite antenna.
Nick sees it now. That is really beautiful. It looks so delicate. Kind
At
of along
what?
the edges.
Wow.
Still? Right now, can you hear it? Can everyone pull their mics just a little bit away from their faces?
Yeah, we've got a breather.
Or
An
mute it.
unidentifiable
That's cool too.
breather. Wow, these sponges are really amazing. This is cool to see all the different ones.
I feel like now no one's breathing.
Everyone's
I've been holding
everyone's
my
holding
breath
their
since I
breath.
said that. I just don't want it to be me since,
All right, full wide, please.
since I made the statement.
Everyone hold your breath.
Oh, man.
Well, it could just be who's the loudest turned up. Nick should be able to control.
Yeah,
Yeah,
and
that's
it wouldn't be.
probably
It may not be on you, but
Yeah, I have the ability to turn people's volumes down really low, but then when you start talking, no one can hear you. So I have to manage kind of how to do that. It's easier if people just mute their microphones when they're not talking. We had a pretty funny comment come in.
Some Darth Vader comments about our heavy breathing. That's okay.
We're just really excited.
Exactly.
Plus, breathing is not that bad.
hey, Dwight, I think we should pick up a rock.
Okay, we're on the lookout. We have two of these. What are they called?
Botroidal.
We have plenty of the botroidal textured rocks.
No step.
We're looking for the
Yeah.
other kind, which
Breathe
don't
enough.
have the
Step
manganese
again, please.
coating on them.
So something flatter. The botroidal is like the knobby,
Yeah.
texture we're seeing with these ones.
So I'm
Zoom
not
in
sure
there,
how
video.
they decided this, but Emil thought once we got up here, we
Oh.
might have
Video,
a
zoom in.
different kind of
Maybe.
flatter basalt.
Yeah. I think these this manganese coating nodules in these botroidal rocks are more common at the deeper depths. So as we get up shallower, we'll hunt for that rock, I think. But it could also be something that's broken off from a steep area that's more fresh. Like these rocks have probably been sitting here like this for millions of years.
Gabby, did you know that before crowbars, crows had to drink at home alone?
A lonely
Okay,
crow
full wide.
joke.
I mean, now that you bring up the crowbar, though,
Oh,
I know. I was surprised you didn't say something, Sam.
right.
I was waiting for you to pipe in. We've only got a couple more dives to get
I
the
know.
crowbar
to
out,
figure out
so-
how to sample a coral with a crowbar. We can't use it on corals, but we might be able to use it on sponges.
Yeah.
Oh.
Break off a little piece.
Or fishes. Nah.
I don't know
You
if
have
that
to,
would
like-
I
work.
would love for you to walk us through your plan there, Gabby.
Yeah. Visualize.
I don't really have a plan. I was planning on picking up the crowbar and seeing
like
where it
like
went.
attracting it to a shiny object, maybe.
Or, like,
Maybe
luring
this
it in.
crowbar is not the
Corralling it.
shiniest crowbar
I
though.
think the best way to use the crowbar to sample a coral is to use it as a peg with Mongo, you know, like just like kind of put it on the floor so you don't have to actually sit down for holding position properly.
I'm wondering if it's our flag. You just plant it at a site while you're sampling, so everybody knows not to come and try and sample your corals.
We're busy
while you're
here.
sampling them.
Because there's so much competition down
Yeah,
there.
Yeah, and in case they don't notice the ROV there.
You know, maybe.
We're just sassing
Zoom in
you.
there.
Who am I to say what they're thinking?
I know. That ROV, that looks approachable, but that that crowbar, oh no.
No way. So we've got some viewers asking about the lasers we use, and we actually just use these to help us know kind of how large the things we're looking at. So it's
Very full
about
wide.
10 centimeters apart between each of those two dots. And a couple people wondering how long we're going to keep diving today. I think we've got another, would it be 12 hours? 16 hours about?
About 16.
16, thank you. Math is hard right now. So we'll be down here for a while. Not us. We'll be back later, but.
Nope.
Yeah, we have to take a pee and snack break.
But then right back.
We got to get our ice cream.
Yep, ice cream break.
Oh, I see.
The robots don't need pee and snack breaks.
No, they don't.
Sam, do you know if glass corals are the same as glass sponges are the same as other sponge types where they can reaggregate if they're separated? Like when I worked with other sponges in the past, you can like pass them through a cheesecloth and break them up into their individual cell components, and they'll reform into a sponge.
What?
Wait,
Yeah.
what?
Yeah, that's the thing.
Wow.
Wait, can you say that again?
Yeah, so like I don't know if it's the same for glass sponges, but with other sponge types, and I've done this in the lab before, you can take them and put them through something to disaggregate them, like a cheesecloth, so they break up into their individual cell components. You can see them only under a microscope at this point, basically, and then you can watch them reform into a sponge, and if you give them a couple weeks, they'll be a visible sponge again.
A couple weeks?
Yeah.
Wow.
But you can start seeing it immediately, but just to the naked eye, it's not as visible for a week or two. But under a dissecting microscope, you can see them within the day already reforming.
But like, they reform, like, not that they grow new sponge. It's they
They come back together.
Oh my God.
I know.
That's crazy. That's really cool.
Yeah.
And I wouldn't be
Wow, look at
surprised
this
if they
guy.
have
Bridge
more
nav, step again, please.
trouble with that, since
Yeah,
they're so
that's
like
what I was
dependent on
thinking.
their silica structure.
Yeah.
But I'm sure they can. Well, I would think they could regrow.
We could find out.
Crowbar one and come back in a couple years.
How can they do that?
Because these other regular sponges, they function similar to kind of what we were talking about with the siphonophores, where individual components are like not their own entities, but they're their own entities enough to where they continue functioning and can come back together. So it's not the same thing as most animals that we're accustomed to, where this organismal structure that is dependent on that shape and form for the movement of food. Individual components of the cells can function and when separate can continue function and then come back together as a whole.
Full wide.
But they like kind of look for each other, or they or they just
It's like a
happen
Power
to attach
Ranger.
to the ones that are nearby and that's it.
I'm not sure if they're only looking for this. Like, I'm not sure if you took multiple sponges and separated them, they would just come back together.
As a one sponge or as several sponges?
Depending on the space in between them.
What if you plant 3 sponges together and put them in the same like experiment?
Well, I
Then
think
you're a
we'd have to.
physiologist.
Yeah. Sounds like you need to do some experimentation. Albert.
Do you want to genetically engineer a super sponge or something?
No. I just want to see. I find
I'm making a sponge that is made of all the sponges. It's an encrusting sponge, it's a barrel sponge, it's a stalk sponge.
it's the sponge. No, I'm just extremely surprised.
Yeah, it's crazy. But these sponges, they're each individual component is, if you look at it like under a microscope, they each have like little appendages that they use for feeding, and they function so much more different than how we're normally used to perceiving animals as functioning. So it works out where they can come back together.
Yeah, it is a little bit.
All right, here's the answer from the literature itself. Study from 1979. Glass sponges can regenerate, but only if no more than 10% of the sponge was wounded. Larger wounds usually cause the sponge to die.
So not as invincible as other sponges.
Wait. This reunion thing that Sarah was explaining, is that regeneration? Or
No.
regeneration is regrowing apart?
Yeah, so that's a bit different than what I was describing. Because what I was describing, they aren't
You don't produce new
regenerating.
cells,
They're just
no?
regrouping.
Regrouping.
They're just regrouping.
In the same shape?
I'm sure eventually. I've never observed them. I think that would take a very long time maybe 6 months to see them get back to a recognizable shape.
Do we know much about like the growth patterns of these deep sea sponges? Like corals down here grow pretty slowly compared to the shallow water corals. Is that the same with sponges? Like, do they grow pretty slowly in comparison, or I don't know if we know, but
I'm not sure.
Most things in the deep sea tend to be slow growing, just because of the cold temperature, high pressure, low food availability.
Ooh, there's an Iridogorgia. I haven't seen one of those.
Where?
To the right. It's out of view now, but there was a little one.
What are you after?
I was just naming the coral there.
Oh, yeah.
Let's look at this guy.
Always a welcome sight.
I know, I love these.
Zoom in there, video.
This one looks
These
so
things?
pink.
There's one really good one we got on the way down. No, like on a previous cruise on the way down, because it was hanging off the ledge, so it was all black behind it. I'm kind of getting bored of them. All right, full wide. Sure.
Bridge nav, step again, please.